Approximately 5 million years ago, our ancestors were just figuring out how to walk on their hind legs and still banging rocks together, when all of a sudden a decisive event would occur, something that would change the human species as we know it. They got a fever. But this would be no ordinary fever, with the retrovirus that caused it inserting its own genetic material into our ancestors' genome, for it to be passed on for millions of years. Meet Jamie. Unfortunately, he's recently been diagnosed with ALS, a motor neuron disease that causes the degeneration of the nerves that innervate our muscles and allow us to walk, talk, move, and breathe. While many of the world's greatest minds are attempting to determine the cause of this disease, it is still yet unknown. However, there is one observation in 97% of motor neuron diseases, and that is the presence of the protein TDP43 aggregating within the motor neurons. This has led to many scientists believing that the degeneration of motor neurons is caused from these aggregates. But despite this, what causes them to form is still unknown. Now, in a cell, DNA gets converted to RNA, and that RNA gets converted to protein. And this is where our retrovirus from 5 million years ago comes into play, as the proteins encoded by the DNA have been observed at elevated levels in affected patients. In addition to this, the treatment that prevent the expression of the viral DNA and the production of its RNA and protein have been observed slowing down the progression of motor neuron disease. This has led scientists to believe it likely plays a crucial role in the development of MND. So, is it the RNA or the protein that is causing the aggregation of TDP43 and potentially causing the degeneration of the motor neurons? To figure this out, we need to first ensure only the RNA or only the protein is present in the cell at one time. Getting the RNA alone is quite simple, as all we need to do is put a protein degradation tag at the end of the viral DNA, essentially indicating to the cell that it needs to destroy the protein the moment it is produced. As a result, only the RNA will remain in the cell. Getting the protein alone is a lot more difficult though. But by jumbling up the code just enough by using RNA's inherent ability to encode for the same amino acid used to produce the final protein, with a different sequence of nucleotides, we can encode for the same protein with an RNA that is just different enough so that it won't behave like it usually does in the cell. As a result, any sections of the RNA molecule that might have interacted with TDP43 will no longer, all while the viral protein remains the same. In doing so, we can then examine both types of cells and determine if aggregation still occurs, and then point future scientists in the right direction towards understanding motor neuron disease and give Jamie another chance.